Hey, it's Mr. Donahue again, this time taking a look at 25.11 stuff, nucleic acids. So our objectives are short. They will be to describe the structure and the properties of nucleic acids. Kind of simple. All right, so nucleic acids, what are they? They're the biopolymers that store our genetic information. So all those chromosomes and such, they're made up of nucleic acids, right? So they're what make up our DNA and have all the instructions for all of our cells. It's kind of crazy to think about it, but our heart cells have the same chromosomes, same DNA as like our lung cells or our skin cells. They all got all this instructions and stuff, and it's made of the nucleic acid. So let's take a look at the monomers, nucleotides. So nucleotides are like the basic building blocks. So if I take a look right here, here's just one nucleotide. It's made up of a phosphoric acid right here. It's also made up of a 5-carbon sugar, and it depends on what sugar you use, what kind of molecule you get. If you use ribose as your sugar, you end up with what we call RNA. And if you use deoxyribose, you get DNA. And that's literally the only difference, only structural difference between RNA and DNA. Because of that change, you get other significant changes, though, like RNA is single-stranded, where DNA is double-stranded. So that's because of the use of a different sugar. And then the last part, which is also very critical, is the nitrogen-containing organic base. So our base right here is C for cytosine. And, those are our 4 letters when we talk about the genetic code. We have G, C, A, or T. And all of our genetic code is used our genetic code is composed of those 4 letters in our DNA. Cool. So taking a look at the nucleotides, we got adenine, we got thymine, we got guanine, and we got cytosine. And then how do they pair up? You see here that they're making these hydrogen bonds. And that's what holds the 2 molecules of DNA together to make that double helix, is the hydrogen bonds between opposite chains. So here I got R, that's my chain over here. Here I got the other R, the other chain. And they link up because of these hydrogen bonds. A pairs with T because of where the hydrogen bonds exist, whereas guanine and cytosine have different locations for their hydrogen bonds, and they will pair up and match with each other. So, yeah. So A pairs with T. I remember the straight lines pair with each other and the curves pair with each other. So G pairs with C. All right, polymerization. It's basically you take many parts and you bind them all together. So for nucleic acids, the OH group of a phosphoric acid, which is this right here, and the OH group of a sugar on another nucleotide are going to react. They're going to give us a condensation reaction that's going to bind them together. So we remove water, and then they are linked together at an oxygen. So if we take a look here, you can see here I got my phosphor group, here I got my sugar, and then again phosphor group, sugar, and I got my organic acid sticking off over there. And you see right here where they're linked at an oxygen from that condensation reaction. Cool. So once everything's polymerized and we got these multiple strands, these 2 strands binding together from the hydrogen bonds right there, it ends up taking on a double helix shape. So you can kind of see like this looks like a ladder, and it's linked together like this. And then what happens to that ladder is that it starts to twist. And that twist is going to give it a double helix structure. So that double helix is saying that we got 2 helixes, and helix is basically a spiral. So if you take a look, you can see it kind of looks like a ladder that's been twisted, right? So boom, you got the different handrails, and they get all twisted, and then you have the rungs, the different legs twisting in between. So it looks like a double helix. It looks like a twisted ladder, all right? DNA replication is really interesting. We're only going to talk very briefly about it. Each strand serves as a template for making a new complementary strand. So remember that the G's pair up with the C's. So first thing we're going to do is we're going to unzip our DNA. So you can see it's unzipping here and splitting into 2 different strands. And that opens up the inside. So you can say, hey, all right, I need to make another strand for this strand. Well, what's going to go with this G? A C. What's going to go with that A? A T. And you can see it's forming the template for how that other strand has to look. And what's really cool, as you can see in this image, the molecular machinery is only gonna make a copy of the DNA in one direction. So it's like 5' to 3' or 3. They have ways of naming directions on this, but basically the molecular machinery is only going to work in one direction. And these strands go in opposite directions. So when we're replicating DNA, one strand can be made continuous as we're unzipping the DNA, whereas the other strand can only be made in fragments because it's going in the other direction. And we get these things called Okazaki fragments, which are pretty interesting. I'm not going to go into great detail, but basically it's going to go like when it's making copies in the non-continuous direction, it's going to go and then it's going to stop. And then it's going to make another strand. So we end up with all these strands that aren't linked together, and then another molecule is going to have to come in and link those Okazaki fragments together. Look it up. It's pretty cool if you're interested. So that's basically it for nucleic acids. So what can you say about nucleic acids? What can you say about DNA? What can you say about its structure and why it takes on that structure? Why are they important? And yeah, so that's pretty much it. I hope you found it helpful. I'll see you in class. All right.